My fever so much. What I heard, taking too much antibiotic not good, ma.
No, you must take this antibiotic for 10 days, or else you may develop the AMR bacteria.
Wait a minute!
What is AMR bacteria?
AMR occurs when the germs undergo genetic changes naturally over time and become resistant to the antibiotics and other antimicrobial medicines designed to kill them. This is due to the misuse of antibiotics and also improper infection and disease control. AMR, I don't want to die. Okay, I eat.
1 week later. Many months later.
How to help this world to fight against AMR bacteria?
Whole genome sequencing (WGS) is a lab method that allows us to determine the order of an organism's genome in just one process. By using whole genome sequencing, we can classify and track AMR bacteria through specific allele profiles, not just phenotypic patterns based on drug classes. This is because whole genome sequencing enables us to have million bases to compare for DNA sequence-based surveillance. Thus, we can have more accurately defined multi-drug resistance.
Illumina is one of the most leading sequencing technology in genomic lab. It offers highest throughput, which produces relative short read up to 300 base pair, with the lowest cost per base. First, the DNA was fragmentized, ligated, and was attached to the glass plate. Next, the DNA fragment was amplified by DNA polymerase. Fluorescently labeled nucleotides are watched across the flow cell and are incorporated complementary to the DNA sequence of the cluster fragment. Fluorescence from the incorporated nucleotide is detected, revealing the DNA sequence. After assembling the data and identifying the genomic feature of the microbes, the next thing to do is to annotate the information. Normally, the data produced from NGS are difficult to assemble. However, we can use bioinformative tools such as metagene annotator to predict the gene on short sequence from uncharacterized metagenomic communities. This software can automatically detect prophase gene and chromosomal backbone prokaryotic gene. Besides, it also can predict translation starting point. Metagene annotator can also be used to analyze the evolutionary relationship. This is because researchers can obtain the information for translation initiation mechanism from the ribosome binding site map output.
A single database of all known resistant genes or mutations should be established to ensure accuracy between different systems and bioinformatic tools. The database should be updated frequently, and minimum standards for addition of new resistant genes and mutations must be strictly established. Here comes the development of the next generation sequencing catalog database.
Meanwhile, the NGS catalog database are particularly useful for researchers from all around the globe in dealing with the AMR spread and growth. By using the data we obtain from the bioinformatic tools, the researcher found that there are some approaches that are useful in dealing with them.
Okay, the first approach is through the identification of the non-genomic signature of AMR from the WGS data.
After done the whole genome sequencing and started to analyze it, the antimicrobial resistance can be inferred by identifying the presence of known antibiotic resistant genes or its variant. The scientists then converts the fixed gene set and their mutations into corresponding binary or real number vectors. Then, subsequently uses them for training machine learning models in predicting newly sequenced samples. The AMR markers can be linked to treatment responses, guiding healthcare workers as they review the patient diagnostic test results and look to make responsible treatment decisions. The pros for this approach are the sequencing of the bacterial genome has been very fast, unlike the traditional lab method which required additional growth in vitro. Hence, this can be applied clinically where the doctors can start effective antibiotic treatment earlier, thereby increasing the quality of care in the era of drug resistance.
Okay, the second approach is through the tracking of gene expression in response to antibiotics.
It is quite important to examine the changes in gene expression of the isolates upon drug treatment. Under stress conditions, for example antibiotic administration to the bacteria, evolution could be obtained to resist the drug to a certain degree. Evolution comes in two ways, which are the mutational resistance and horizontal gene transfer. By understanding the biochemical and genetic basis of resistance, scientists could design strategies to cushion the emergence of resistance by having some device innovation therapeutic approaches against those AMR organisms. Doctors then can administer antibiotics more responsibly, taking measures to cut down on overuse. However, the experiment is still complex and a long time is required to generate and analyze the data, so this approach is unlikely to be employed in a clinical setting that soon.
Okay, the third approach is through the identification of AMR mechanisms from the metabolomic data by using the benefits of metabolic profiling.
This is because metabolism plays a central role in bacteria to mediate early response against antibiotics and demonstrate antimicrobial resistance. These approaches gives a new perspective on the understanding of antibiotic action and response of bacteria to the drugs. Also, lays the foundation to improve the drug treatment regimens and optimize combined therapies. On the other hand, it is still far away from being easily translated into clinical practice since not all species are well studied in terms of the metabolic pathways.
In a nutshell, researchers can utilize the bioinformative tool against the outgrowth and spread of the antimicrobial resistance.
This can be done by collecting the data from the sequencing technology such as Illumina and then upload data to the database such as NCBI.
And thus, the data can be utilized by the researchers from all around the globe to identify the genomic signatures of the AMR from whole genome sequencing data.
Track the gene's expression in response to antimicrobial and
Can track the AMR mechanism from the metabolomic data by using the advantage of metabolic profiling.
I'm Jianzhou.
I'm Jizun.
And Qianfeng.
That's
That's all from us.
Thank
Thank
you.
you.